So as we discussed last time, the whole point with MRI and fMRI is to try to look at the brain, so to try to look at brain tissue and in particular brain function and neuronal activity. And we try to look at this by using the rates at which protons dephase with respect to each other. So you have to keep in mind that when we do MRI and fMRI, we are studying neuronal activity in a very indirect way. As opposed to actually seeing neurons as they fire, as they engage in activity, as they get excited because some event happens, we are looking at the effect that neural activity has on the rates at which protons dephase. Okay, so again, fMRI is an indirect way of studying neuronal activity. So it's very important that we understand what is the link that unites the neuronal activity that we're trying to understand with the actual thing that we're measuring, which is nothing but the rate at which protons dephase with respect to each other. So T2 or T2 star relaxation. So it's very important to spend some time on trying to link these two things. In fact, this is what we're going to do half today and half in the next lecture, trying to tie a link from neuronal activity all the way to protons dephasing, and how the former, neuronal changes in neuronal activity, affect changes in the latter. So in the rates at which protons dephase with respect to each other, which again is what we are measuring. So let's do this. Imagine you take somebody, you put them inside an MRI machine, and you just have them stare at a blank screen. So in the sense, you're putting your participant in a rest condition or a basal condition. All I mean by this is that you're not engaging them overtly in any particular task. So obviously their brain is there's a lot of activity going on all the time, but it's all, for our purposes, background activity. It's all basal activity. It's not related to one specific type of task or mental activity that we will ask them to perform. Okay? So you take a participant, put them inside the MRI machine, have them stare at a blank screen. Okay? I'm going to refer to this as rest. So a condition where we're not asking our participants to do anything, as opposed to the condition which we'll refer to as task or active, where we overtly ask our participants to engage in something. So here we are, participants inside the MRI machine, staring at a blank screen, so they are in this rest condition. If we could look at the tissues at inside and surrounding blood vessels, for example, capillaries and small veins, we would see, obviously we would see a lot of hydrogen protons, which are depicted again here in blue, and these are inside water molecules. And then we would probably see some amount of oxygenated hemoglobin in red, and some amount of deoxygenated hemoglobin in purple. And remember, the deoxygenated hemoglobin is paramagnetic. So if you were to think of this in terms of what is the Larmor frequency at which each of these protons is resonating, the presence of deoxyhemoglobin makes it such that these protons here, because of the deoxyhemoglobin, are probably resonating at a slightly different frequency than the protons that are nearby only oxygenated hemoglobin, which is diamagnetic and does not change the magnetic field. So the net result is that protons in this small piece of tissue, in this particular moment, given this amount of oxygenated and deoxygenated hemoglobin, will probably dephase rapidly. So the T2 star relaxation will probably be quite rapid. So at TE, whenever we look at how strong the signal is, whenever we, as we said last time, snap the picture, to speak slightly improperly, probably won't be very strong, because different protons, because of the deoxyhemoglobin, are resonating at a different frequency. Therefore, they're going to lose coherence and dephase fast. So this is at rest. Now let's see what happens if we trigger activity if we trigger neural activity with some kind of task, overt task. So our participant is still inside the MRI machine. Now we show them a picture of a face, maybe. And so I'm going to refer to this as task or activity. So we show our participant a picture of the face, and now all the parts of the brain that are involved in recognizing a visual stimulus, in processing it, and recognizing it as for what it is, a face, all those neurons will suddenly have a burst in activity. Okay, so neuronal activity in a particular part of the brain, the one that is important for processing visual information and faces specifically, all those neurons will have a burst in activity. What happens and how does it change the MRI signal? Well, as neuronal activity increases, there's an immediate metabolic response. So there's an increase in glucose and oxygen metabolism. As a result of the increase in glucose and oxygen metabolism, there will be an increase in cerebral blood flow and cerebral blood volume. And don't worry about this. We're going to spend a whole lecture on these details. For now, I'm just trying to give you the broad overview. Now, as a net result of the changes in cerebral blood flow and in cerebral blood volume, what happens is that the levels of oxygenation inside the blood and the tissue surrounding the blood vessel increases. So suddenly, the ratio of oxygenated to deoxygenated hemoglobin has changed in favor of oxygenated hemoglobin. So to think of it with the same picture as before, during task, what happens? Well, look here. Now there is much more oxygenated hemoglobin in red and much less deoxygenated hemoglobin. So what's going to happen to protons in terms of their dephasing? Would you imagine that they dephase faster or slower compared to before? Well, think of it this way. If there's less deoxygenated hemoglobin, it means that the magnetic field is a little bit more even, right? Because deoxyhemoglobin changes the strength of the magnetic field. So if there's less of it, it means that now all protons are resonating almost in the same magnetic field. So they're almost all resonating, obviously at the same gyromagnetic constant, but also inside the same B0. So this means that they're all going at a much more similar Larmor, they're all resonating at a much more similar Larmor frequency. So if they're all resonating at the same frequency or at a more similar frequency, it's going to take longer for protons to dephase, right? The magnetic field is more uniform because there's less deoxygenated hemoglobin. And as a result, protons will resonate together for longer because they're all going at the same pace. If they all go for longer, if we wait for the same TE and then snap a picture, how will the signal be? They'll be stronger because protons are swinging together much more than before. It takes longer for protons to dephase with respect to each other. So if we wait, let's say 10 milliseconds before we snap a picture, now the signal will be stronger compared to when we took the picture at rest. Here there was all this deoxyhemoglobin making protons resonate at different frequencies with respect to each other. Here there's much less of it, so protons go a little more together. So it takes longer for protons to dephase. And so when we take our picture after, as I said, let's say a TE of 10 milliseconds, the signal will be stronger. And this is the whole idea behind fMRI. And with this mechanism, I can see that certain parts of the brain, in response to seeing, for example, a face, will now have a stronger T2 star signal. So think of it this way. We can reverse the inference. Let's say that we have somebody look at a picture, and suddenly I see that there's some part of their brain compared to before that has a stronger BOLD signal. Okay, so now we are up here. If that part of the brain has a stronger BOLD signal, I will say, it means that protons, compared to baseline, compared to rest, are resonating at a much more similar Larmor frequency compared to each other. Now, why would they be resonating at a more similar Larmor frequency? Well, it must be because the magnetic field is more uniform. What makes the magnetic field more uniform? Well, in our case, the one big variable is changes in blood oxygenation. There must be more oxygen, more oxygenated hemoglobin, and less deoxygenated hemoglobin. Okay, so what causes an increase in oxygenated hemoglobin and a decrease in deoxygenated hemoglobin? Well, the cerebral blood flow and the cerebral blood volume response to changes in glucose and oxygen metabolism. Okay, what changes the glucose and oxygen metabolism? Well, a number of things, including neuronal activity. Okay, so when we do an MRI, an fMRI experiment, and we say, oh, look, during my task, the signal was stronger here, and it was not as strong when my participant was doing nothing, then this is the chain that unites seeing a change in the BOLD signal all the way down to neural activity. All right? Finally, you should appreciate that fMRI is a relative tool in the sense we don't interpret it quantitatively in terms of how many protons are there that are resonating together. What we do is we look at how strong the BOLD signal is under one condition, rest, and under a different condition, task. So every time we are looking at the BOLD signal, we're always looking at it in a relative way. For example, in this image here, at rest, whatever part of the brain I'm looking at doesn't have a very strong signal. Whereas during task, so for example, while looking at pictures of faces, my signal is much stronger. So relatively speaking, when somebody engages in a given mental activity, for example, looking at a picture of a face, that this part of the brain is having a stronger BOLD signal. And that, as I said earlier, is how we tell that a part of the brain is related to some specific mental activity. So conventionally, we often break down this schema, this chain of events, as the physiology of BOLD. So what happens from a physiological point of view in response to neuronal activity. So neuronal activity increases, and that leads to a certain change in metabolism and a certain change in flow and volume, and therefore a certain change in oxygenation. And we're going to discuss this next time. And then there's the so-called biophysics of the BOLD signal, which is if the magnetic field becomes more uniform because there's more oxygenation and less deoxygenated hemoglobin, what happens to the magnetic field? How does it change? How does it become more or less uniform? And how does that change the rate at which protons dephase with respect to each other? And this we refer to as the biophysics of BOLD. So what I want to do today is focus on the BOLD biophysics. And the next time we will look at the BOLD physiology.